Okay, so hi everyone. If we could just settle down and we'll start the presentation here. So, orchid viruses. When I was starting out with orchids, this wasn't really something I was all that aware of, and I stepped into working in like a large collection in a conservatory-like setting. And we had around 500-600 plants. And I had looked back on like our accession sheet for the orchid collection, and I had noticed that like there was a column for orchid virus testing, and like there were some negative tests on there. And I didn't think too much about it at the time because there was so much else to do. But over the years, like not paying attention to new plants that come in that might potentially have orchid virus led to some problems down the road. I brought in some sample plants right here just to show off what symptoms look like on some different types of orchids. All of these plants here tested positive, and it's been something that I've just been battling in the greenhouse for years now. I've even gone through and tested the entire collection, and still, like, it's on potting material, it's on the floor, it's on benches. It's something that once it starts spreading in the collection, you have to be really careful about your sanitation procedures. So I'd just like to give everyone a little prep and a little advice about orchid viruses so that you could start out and stay clean. So this is just a little COVID humor, because plants are not that much different than people. They do suffer from viruses. Let's see. So why do we even worry about orchid viruses? Like I said, orchids are susceptible to diseases, pests, fungal pathogens, bacterial pathogens, and viral pathogens. There's something that you don't really think about when you get a plant. Like, you're like, oh, okay, it gets a fungal symptom that you see, it gets a bacterial symptom, but orchid viruses are unseen and they're very prevalent out there. I believe a lot of plants don't show the symptoms, like they're asymptomatic. And you can have a beautiful plant in bloom, healthy, and then a plant right next to it that has genes that are susceptible, and it'll start showing symptoms once it starts spreading. So viruses tend to weaken plants. They don't necessarily kill them. So a lot of times you'll have a healthy plant and then a viral plant right next to it, and it could be the same exact plant, and you'll notice just a decrease in bloom quality, quantity, plant vigor, the amount of growth that it puts on every year. A good example of that is this one Callie over here that I have. You see that all the older pseudobulbs are quite large and fully developed, and then all the newer growths on this plant are smaller than the old growths. I mean, it could be partially because it's woefully underpotted at this point, but the viruses reduce plant vigor. Why else is this a problem? Why do we grow orchids? It's for the blooms. That's our crop, that's our harvest. So when we assess our plants in our collection, whether they're worth keeping or not, it's the blooms. That's the whole point of why we grow orchids. And if you're reducing the quality of your blooms, that's the biggest impact that orchid viruses will have on your collection. As I mentioned, some plants are more tolerant than others. You can have asymptomatic plants in your collection, and that's where you run into problems when you don't have good sanitation between plants, and you unwittingly spread them between plants that have good genes and can like handle an orchid virus, and a plant that is very susceptible and shows severely debilitating symptoms just visually. Let's see. So this is one example, a healthy plant versus a virus plant. So left flower is a healthy plant, the right, exact same flower with virus. This is Braytonia Charles M. Fitch. I actually brought that one in at the last meeting, the healthy plant. And then I have the virus plant right here. I wish it still had its flowers, but it was really interesting what the virus did to the flower itself. You can see that pattern on those petals. It's fragmented, it's like very convoluted. And when it first opened, like the plant was healthy. It wasn't showing any symptoms of decline, no reduction in vigor. And after repotting, I'd noticed that the plant, the older leaves were starting to look poorly. It was starting to drop some older leaves. The new wasn't really pushing too much root growth. And then it pushed a flower spike, and that's what the flower looked like. And I'm not thinking that it was during the repotting that it got this, but it was probably pre-virused, and then the shock and the stress of being transplanted led to a severe expression of the symptoms. So not only was it the floriferousness of it, the quantity of it was affected. It was like a plant next to it that was healthy had way more blooms on the same stem compared to the plant with virus that had only few blooms on the stem. And the flowers that did come out, they were thicker in appearance and distorted. And so there's really no reason to keep a plant like this in your collection. Like, it's not something that's going to get better. Orchid viruses, there's no cure for them. They're not like people where you have an immune system. They do have an immune response. That's why some of them don't express the symptoms as severely. But the virus is always going to be transmitted. It's always active in the plant and in growing tissue. The plant is trying to outgrow it. Some plants can outgrow it faster than others. But even a very healthy plant that has virus can still spread it to any plants around it. All right, so symptoms are diverse. We grow a lot of different kinds of orchids. And the symptoms appear differently on different plants. A lot of this has to do with the genetics of the plant. You could have one type of Cattleya hybridized with another type of Cattleya, and like depending on the vigor of that cross, some plants show severe symptoms more severely than others. So presentation, plants could be asymptomatic, and that's a very common thing. Just because it's symptom-free doesn't mean it's virus-free. The only way to know for sure is to test for virus. And it's important that you familiarize yourself with what the different symptoms look like so you can distinguish between pathogens, diseases, and viral symptoms. So if you have a plant in your collection, it doesn't look good, you see strange spots, strange markings on the leaves, something isn't right, you want to know whether it's environmental, whether it's a plant pathogen, whether it's an insect damage, whether it's something you did, an animal or something, or whether it's a virus. And just being familiar with what it looks like, after a while you can walk into a collection of orchids, just look at a plant and be like, it's probably virus, just based on the physical presentation of symptoms and the type of plant it is. There was a really good article, I think it was January 2023 in the AOS magazine, about orchid viral symptom presentation. And like they just had a really good photo gallery of different symptoms on different plants. So what are we talking about when we're talking about viruses? So at this point, I think there's around 30 different viruses that are known to infect orchids. So that doesn't mean they're all common, but different plant viruses can jump between different types of plants. Usually there's host specificity. So one virus, like CymMV mosaic virus, or I believe it's, yes, it is odontoglossum ring spot virus, is TMV. So tomato mosaic virus. Tomato mosaic virus is odontoglossum ring spot virus. It's the same virus on a tomato, and it probably jumped onto orchids at some point, and now that's one of the most prevalent diseases. It just has a different name because it's on an orchid. So of the 30 that are currently recorded to affect orchids, the most common ones are CymMV mosaic virus, odontoglossum ring spot virus, cucumber mosaic virus, orchid fleck virus, and one that I ran across a few times but I hadn't run across before was capsicum chlorosis virus. Orchid fleck and capsicum chlorosis virus, I believe those are two that are more common on Phalaenopsis, and they've been introduced through Japan and through the large Phalaenopsis trade, the moth orchid. But the two most common orchid viruses that you're going to run across in your collection are probably CymMV mosaic virus and odontoglossum ring spot virus. Because they're both very stable, and they're mostly transmitted by mechanical means. So like when you're working with your collection, we are the ones that are spreading these viruses. They're not found in nature. These orchid viruses are just prevalent under the man-made conditions that we produce. And since we're constantly damaging our plants when we're working with them, they're spread very easily between one plant and another. I think, I was looking it up, and I believe like odontoglossum ring spot virus is like heat stable to like 95 degrees. And it has like an in vitro life of like 10 years.
You were saying how it's passed by us planting, potting, whatever.
Mm-hmm.
Have you ever used or know of Physan 20, which kills all that stuff, like wash your hands? It says wash the table. Would that help?
So we'll come out near the end. We're gonna talk about sanitation and sterilization. We're gonna talk about Physan. That's one of those sanitizers that really doesn't do too much for these two viruses. They're good for a lot of other things, but we'll talk about ones that really do work.
All right.
Yep. So as I was saying, those two viruses, CymV and RSV, are mostly spread mechanically. by people, but then a lot of the other viruses are spread by insects. So you have some that are spread by aphids. I believe cucumber mosaic virus is mostly spread by aphids. I believe orchid fleck virus is mostly transmitted by mites. And Capsicum chlorosis, I believe it's mostly by thrips. I have a slide for each one, so we'll make sure that those two, and I'll talk about those pests. So it's important in those cases to manage the insect pressure in your greenhouse and manage weeds in your greenhouse that host those insects. We'll talk a little bit about that when we get to them. But the two most common ones, we're going to go over proper sanitation and handling practices for your collection.
And these aren't just greenhouse specific, they could be in a homegrown windowsill environment as well.
Yes, so yeah, I mean, all orchid growing conditions are favorable for those two viruses. Yeah, it's not, I'm talking specifically in our greenhouse collection, but you could have like 5 plants on your shelf in front of the window and one plant has it, and you're working with your plants and you spread it between all 5. They're easily transmitted. So these are the 2 viruses that you'll find. I just put down the viral group that each are in. And it's interesting because like Tibamo virus, it's from tobacco mosaic virus. Potex virus, I forget what that one comes from, maybe potatoes. But there's bean yellow virus, and like they spread from common agricultural crops into our collections. Yes, Dave?
And I don't know how true it is, and it's not as appropriate anymore because not as many people smoke. But back in the 60s and 70s, the word was out that if you smoke, make sure you sanitize, and don't smoke and touch your orchids, because you spread tobacco mosaic virus, which is odontoglossum wings virus from the tobacco.
Oh my gosh.
It's very common
I
in
don't know
commercial
how
tobacco.
true,
And
you know,
yeah.
that is, but that was commonly practiced.
Wow.
Yeah, they were very paranoid when I was going to Cornell in the research greenhouses about CymMV. You couldn't smoke anywhere around the research facility. You had to dip anything that went in the greenhouse into a sanitizer, TSP. And it was a serious thing. I remember a horticulture club was growing ornamentals in one greenhouse and they kicked us out of the facility because they didn't want any plants that might host CymMV. So we couldn't even, the horticulture club couldn't be in the building. It was pretty serious.
We could decimate a whole collection.
I mean, if you have a genetic, like something that's rare, and you have genetic research that you're doing, and you get a virus in that collection, it's all worthless. So, CymMV, it's probably the most prevalent orchid virus you're going to run across. Everything on this table, well, minus the 2 that we haven't tested yet, has CymMV. It's not found in the wilds, just because it's mechanically transmitted and not vectored by any insect pathogen. It's through contact of contaminated materials, potting material, your hands, pruners, media, pots. It's usually expressed as a mosaic and a necrosis of the leaf. So that's why I brought some plants in that you can see them up close and in person. It's hard from photographs to really get an idea of what it looks like. You have to see a lot of leaves and see like how it's expressed in those leaves to get a sense just by looking at a plant how it's expressed. The other one, ORSV, also known as tobacco mosaic virus, like we talked about. It's very persistent. Like you could smoke that cigarette, and I mean you're burning the leaves to ash, and that virus is still active. It's very heat stable. It's very UV stable. Well, I'll have a slide where they talk about how hard it is to kill this virus. I think it's like 100 times, you need 100 times more UV radiation to deactivate the viral RNA than you do to kill like common bacteria on your table. It is very stable. And ORSV is mostly responsible for color break in flowers. So one of the symptoms in Cattleyas is to get streaking or color breaking in your flowers. And of course that ruins it for display purposes. Interestingly, well, that one doesn't have ORSV, but specific to ORSV, you will see color break in the flowers. And that was the picture we posted on Instagram and on Facebook of that Cattleya flower with those flecks of color running through them. Sometimes it can be genetic. There are some flowers that have streaking and spotting in them that aren't virus. And sometimes you get it and see it in Phalaenopsis a lot, the color break. And that's genetic. But you have to test to know for sure. The leaf symptoms are similar to CymMV. The lesions are sunken, necrotic, chlorotic. Usually it's a little bit more of a ring that you'll see. And it's expressed differently on cymbidiums than it is on cattleys. All right, so like I was saying, it's important to know what the viruses look like on different types of plants because they're going to express differently. And it'll express differently in different life stages. So like you've got these orchid seedlings in a flask. Usually you won't see the viral symptoms expressed in such a young plant. So the only way to know is if they were to test and index that collection. That seller would have to test a plant or do like a little sample of each little plant test to know for sure, because viruses can't be transmitted through orchid seed, but if there's any other material in the pod, like there's all those fibers, there's the husk, and if there's any material from the parent plant that gets mixed in with the seed and then gets cultured, that's a flask of orchids. The viral particles can infect the new plantlets as they're growing out roots and new leaves. It might not infect all of them, but it will likely infect some of them. So the other thing to know about symptoms is that if you have multiple factors overlapping, like different viruses, the same plant can be infected with multiple different viruses, and that's severely debilitating. And it looks really strange on a plant. I've had plants that look, it's just like they crash. The plant just crashes. I mean, it can handle one virus, but then when you add something else on top of it, it's debilitating. So this one at the end right there, that cattleya has both ORSV and CYMV. When it flowered the last time, the flowers were so twisted and convoluted that it didn't resemble anything like the original plant. I'm surprised it's still putting on so much new growth. So genetics make a difference. In that case, that is a very vigorous grower. I mean, it has nice tight growth, lots of lots. They won't spread through the air.
Oh, that was good.
Sorry.
Social distancing.
It has lots of new growth, so it's a really strong plant. So it's fighting through the virus, and it's even going to flower this year. But other plants will just like shrivel away, like this little cattleya up front here. Like it's just starting to shed all of its older leaves that it would normally hold on to. So it's losing vigor, and eventually it's going to kill this plant.
Hey, Christian, I've also found where a fungal or bacterial infection will make a virus express itself.
Yes, any stress.
Any stress.
It could be environmental. It could be pathogen like that. It's life stages of the plant. Like these seedlings here, you most likely won't see viral expression yet, but you can start to see it in young plants. You'll usually see it in older plants, like the plants that have been in a pot for a while. The EC is starting to rise. The pH might not be exactly where it wants to be. There's a lot of other pathogens. Those leaves have like, I believe they have like bacterial spotting on the leaves. So like that plant's starting to get under stress. It's slowing down. It's not as vigorous as when it was young. The symptoms are probably going to be most severe in a large plant like that. I usually see it after repotting, and I've started just testing before I repot because you don't want to be spreading any virus around your potting supplies on your table as you're working. So pattern recognition. So on the left, we have some very common CYMV, and on the right, ORSV. So you can see that chlorotic streaking on the leaves. I believe that was some type of Oncidium or Basidium. That is extremely severe. Once it reaches that point, you know you have something wrong. Usually it's just a little bit on the newer growth. Like I have some dendrobiums here, and it's subtle. Like I didn't know they were virus until I was getting ready to repot and I took a test. And then I looked closer and I was like, okay, what I thought might have been a nutrient deficiency, a little bit of chlorosis on the leaves, just a slight veination, slight bit on the veins. But then once the plant gets older, it gets stressed, that'll get really severe. So once initially it starts out chlorotic, it'll eventually become necrotic, and you'll start to see sunken streaking on the leaves. And that's where most of these are at this stage, is necrotic spots and streaks along the veins. They're monocots, so their veins run parallel to the leaf. So ORSV, it's that color break. You see all that like white, and there's a little bit of a darker splotch in the center. There's a little streak of white here. There's another streak of white. That's ORSV flower break. It can be more severe to the point where the flower is twisted, doesn't look like the original flower. There are some plants that have that in their genetics. You could test them and it's not virus. It's just how they behave. Sometimes when you mess with hybridization in the ploidy level, you'll get weird floral symptoms like that. But in this case, this popped up, and I was like, okay, plant was asymptomatic, time to test it. ORSV, typically it's like a purple, a dark brown to reddish purple ring on the leaves with a slight chlorotic halo around it, will eventually become necrotic.
Is it an actual ring, like a circle? Or?
It won't necessarily be the ring, but it'll be like sort of wavy, marginally.
Oh.
They call it ring spot virus because it looks sort of, it's a enclosing margin to a central region, expanding boundary area of the virus. All right, so CymMV. So again, those chlorotic to necrotic streaking to spotting that progressively becomes worse. On Cattleyas, it starts out as a very subtle chlorotic streak. You'll just see a little bit of chlorosis in the leaf. You won't really notice it unless you're looking for it. The leaf might be slightly distorted. It doesn't develop perfectly. And then as the plant becomes stressed, like these plants have been in these pots for a little bit too long. So like they're getting ready to be repotted. The media in there is probably broken down. There's probably very few roots that are still in the pot. It's stressed. So now the symptoms are becoming more severe. And then same case with the Presidium. Usually they don't become necrotic on these type of thin-leafed orchids because they'll drop their leaves before it reaches that point. It just looks like natural senescence in the leaf because those types of orchids drop their leaves more frequently than Cattleyas. It's just that Cattleyas, they hold on to their old leaves. It's an energy reserve. There's a lot more invested in that leaf, so it'll develop those necrotic spots. On flowers, on specifically Cattleyas, you'll sometimes see this necrotic development on the flowers as they age. The flower will open fine, and then as it ages, you'll start to see necrosis and like streaking in the flowers. I had a really good example of it on a Potinaria this past spring. I repotted it, and it was just getting ready to bloom. The flowers opened, and it was necrotic after 2 days. It shouldn't happen. Sometimes you get botrytis that'll lead to spotting that looks very similar to this. But just pay attention to the timeline that it happens. I mean, sure, if you misted your flower in the evening or something in an environment where there is botrytis, it might look like that the next day. But it's going to be more streaked along those veins, the major veins of the flower. I wish I'd taken a picture of the flower that I had. It was almost like someone had taken a paintbrush and painted streaks down the center of the petals.
Botrytis is little black dots.
And it's, yeah, we'll have more look-alike photos after this. And it doesn't, it's very different, yeah. And eventually the flower will just drop. The whole flower will become necrotic. So on different types of plants, here we have a dendrobium. I brought this one in. I was just thinking it was like an anthocyanin overexpression because it was in bright light. But apparently that is something that this dendrobium does when it has viral symptoms. And it was also losing vigor. Like all the older leaves were dropping off the old canes. It had some buds that had aborted at the base. There were multiple new growths, and I was like, why is this plant just not doing well? So I was getting ready to repot it, even though it was still freshly repotted. So I tested it, and it had CymMV. And this was a plant that had not been in the collection for a year. Like I had bought this commercially from Calipana Tropical, so I don't know if it came in with the virus or if it got it in my collection. Some big growers might not test everything that they're sending out. And then
Most don't. Most growers just
most won't because it's too expensive.
Yeah.
Could they?
And if it's an old orchid grower, like someone that's been around for a long time, it could just be in their greenhouse. And not all the plants are affected, but some get it, and they get sent out, and they get in your collection. So whenever something new is coming in your collection, you should like segregate it. We're going to talk about like the whole procedure of what you should do in your greenhouse, but I did not because I thought these were clean. And I tested everything in the greenhouse, and I was like, I don't know. Want to spend $4 on top of whatever I already paid for the plant? Well, apparently I should have. So you see on this one, this is another dendrobium, same grower, same time I bought them. There's a little bit of chlorosis and it's a little bit distorted. The leaf is a little wavy where that chlorosis is. Same thing, it's dropping a lot of the older leaves on those canes prematurely. One new growth popped up, but the others all aborted. The plant just wasn't very vigorous. And really, when I saw these, most cane dendrobiums like this are taller and bigger. These are really stunted and small. I thought it was just breeding and newer ones, but these both had CymMV. What's going on out there? So you can look at those a little bit more. I found them on the table here. And it's good to look at them up close because it's hard to capture that chlorosis on the leaf in the photo sometimes. Okay, so on that Braytonia that I showed earlier, I've got it here with me now, but it's that just like strange pixelation of the flower. The spotting on the flower is just distorted. There's a little bit of chlorotic streaking. Okay, this maybe could be fungal at the leaf tip, but it's all the old leaves that are just starting to drop. So there's chlorotic necrosis that leads to premature leaf drop. Here's some streaking, like that necrotic streaking and spotting on in the veins. It's also on these protective sheaths that usually come out with a new growth. And you can see on the pseudobulb itself has chlorotic streaking. And the bulb is kind of distorted. If you look at it at the plant, it's hard to capture it in the photo. The bulb is just, it didn't develop the way all the others had. It's a little bit wobbly and a little bit wonky looking. And so immediately, as soon as I saw that it was starting to express this, I kind of knew it was virus, and so I tested it. In a Cattleya, okay, now we're on to ORSV. So this is just up close at that flower break. So this is another one. This is really funny. I was looking back through old photos. I'd visited an orchid nursery, and I'd taken a lot of photos of plants and flowers, and I was like, huh. It's got orchid virus. When I went to visit, I didn't even know when I was taking the photos. But it's just like that streaking and flecks on the petals. And it causes variegation in normally solid colored flowers. So that's where if you know what the flower should look like and it's got variegation in it and it shouldn't, it's a virus, it's ORSV. It could be mild, it could be severe, you might not even notice it. It might be just subtle. It might actually go well with the flower. It might be so symmetrical that you don't realize it's the virus. But yeah, so this is one hybrid that I have here. And I had multiple plants that had shown this, and I didn't take photos of them. And you know, you get into that, you just throw everything away so quickly that you don't record it. All right, and then on the leaf, the symptoms are those necrotic, sunken spots. And it's usually a ring that can be outlined in dark red surrounding a chlorotic interior. And it's also that streaking along the veins. Same thing. It's just slightly different in its purpling and the chlorotic interior. And I brought that plant in here. So then we move on to CymMV and ORSV together in the same plant. And it was very severe in this plant. You could see that chlorosis. As that new leaf was emerging, it didn't even go necrotic.
So that's on the new leaf?
Yeah, that's on a new
Yeah.
leaf. And it's so severe that the tissue didn't even develop. It's just thin and papery and necrotic. On the pseudobulbs, you could see the ORSV symptoms. Those rings, those purpling rings. And you can see on all the new growths, there's the streaking. On the older leaves, it's become necrotic. But all the new growths show that chlorosis. Older leaves show necrosis. You'll see sometimes on the bottoms of the leaves a little bit of spotting as the chlorosis progresses straight through the leaf. It's usually on the upper leaf surface, and then as it progresses, it'll start eating through the leaf basically. The leaf just starts, the tissue starts dying. You can see the halo on all those old pseudobulbs, that ORSV expressing. And then here's just more close pictures. That's in good lighting what that plant right there looks like. And just that mixed symptoms of two different viruses together. It's really taking that plant down. And you know, as I was saying about like stresses that induce viral symptoms, this plant, I hadn't touched it in forever. It's an old plant, hasn't been repotted in like 5 years. Doing extremely well, hanging, isolated by itself. I wanted to display it. And it had all these roots on the bottom, so it didn't sit nice. So I went snip, 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 sheared everything off flat at the bottom so I could sit on the table nicely and display it. And then I must have just taken that pot, maybe I put it on the ground, and there was something on the ground. There was viral particles somewhere in the greenhouse, on a bench, in the gravel, on the crate that I used to move the plant. And it got both viruses. Because when I tested this plant, 2022, it was clean.
Oh, wow.
And that's in a year and a half. All the last batch of flowers, that was the first sign that, oh, there's something wrong.
And snipping the roots open up the tissue.
Yeah. Because usually when I go in and I'm trying to take off old flowers, I'm pretty clean about it, sanitizing my pruners and making sure that I'm not cross-contaminating between plants. It was really, I know what it was, I know what I did and how that spread. And I feel really bad because it was a really nice plant. All right, so after the two main orchid viruses, there's other viruses that aren't as common. But once you go through, well, in my case, once I'd gone through all my plants and tested them for RSV and CYMV, and there's still symptoms that look viral in the collection, it's time to go to the next step and to consider what other viruses it could be. The cucumber virus group. So it's from cucumbers, cucurbits. This one, it's transmitted by aphids. So if you have other plants in your greenhouse, and I know how this got in the collection because we were growing gourds and pumpkins outside the greenhouse, and those would get aphids. And then the sides of the greenhouse roll up, the aphids come inside and feed on your nice tender orchid buds, and they transmit cucumber mosaic virus. And I mean, the most notable symptom of this one is chlorotic streaks on the leaves. And that's on a path. And it's distorting the foliage too.
So that's a path.
Yeah.
So it's gotten it.
So this one, I have to buy the test kits for this. Because like, I have to start testing the whole collection for it now. Because like, paths are not something
I
that
thought
get,
paths were pretty resistant to virus.
they mostly don't get virus. It's not that they're resistant, it's that they don't get them because we don't do as much mechanically with them. When you're dividing a path, you don't usually cut them apart like you hack a cattleya apart. Usually the roots, you're not cutting roots usually, you're teasing everything apart and then breaking plants apart physically. And they grow so slowly, you're usually not going in there, like when you're cutting apart that plant, it makes a big mess and there's a lot of plant material everywhere. And you're really injuring that plant. Paths, you don't so much. So this one was vectored by an insect. And they like to go after buds. So like in your greenhouse, it's important to control weeds. If you have like, or outdoors, in a garden, if you bring your plants outside, take into consideration whether you've got aphids in your garden. Do you grow squash, cucumbers? Like there could be virus in those plants that could be transmitted to your orchids. This is a frag, like two plants that you would never think would get viruses. That was from a flask that I got it. It's not even an old plant division. And that's a compot. There was a bunch of old plants in there and a few of them had it. Yeah. And it's notably, it's that chlorotic streaking in the leaves. I don't know, sanitation. Orchid fleck virus is another one. This one was first discovered in Phalaenopsis, in mass-produced Phalaenopsis. I can't remember the timeline for this, but like in Taiwan, they were testing, I think it was like 2015 or something. It's fairly recent on this one. But this one, another insect-vectored virus. This is by mite, Brevipalpus californicus. So one of the things to do, they would do to discern whether maybe you have this virus in your collection, is check the leaves under a dissecting microscope to see if you have these mites. Because if you have these mites, there's chances that you'll have the virus. I didn't see very much yet about what the symptoms look like in other orchids, because it's mostly Phalaenopsis that they focused on. It can happen in all orchids. But it's mostly chlorotic flecks, spots, and stripes that eventually become necrotic. So they'll start out as small little chlorotic spots. That will eventually, the necrosis progresses and the spot expands, becomes papery and glassy in the center. And you can see some photos of plants that are just covered. It's mostly on the older leaves. The new growth is cleaner, and the older growth shows these spots. And it can be severe on some plants. It looks kind of bacterial almost, but the difference here is that it's not wet and water soaked, it's dry and papery. All right, so another one, capsicum chlorosis virus. This is another one that is transmitted by an insect vector, by thrips. You could look up what specific type of thrip it is. I'm never good. if you showed me 50 different thrips, I'd never be able to pick this one out. But it's a tosporovirus. But the interesting thing about this is that it's not systemic, it's a local infection. So they've done testing on these leaves, and they've tested the chlorotic region around that halo, and it's positive. But you could test another leaf on the same plant, and it doesn't have the virus. So you could theoretically prune it out if you were really careful, and you caught these bullseyes. It looks very fungal. That chlorosis starts out as a little spot, and then progressively those rings expand to cover the whole leaf, becoming necrotic as it ages until it takes out the whole
If you
leaf.
allow the leaf to die and die
Yeah.
off, would it spread to the other leaves?
I don't know, because they say it's mostly transmitted mechanically by thrips. So the thing about CYMV and RSV is that they're so stable. These viruses outside of host or plant tissue aren't as stable. So like the chances of you transmitting this, you take a scalpel, you slice through that plant tissue, you get that sap that has all the virus on it, you put it down on the table, I don't know how long it would live. CYMV, it could live 10 years. Or ORSV. But this one is a newer one. I can't remember when they first discovered it, mostly on Phalaenopsis, again in Japan. Like I said, that it's not systemic. And I don't believe there are any viral test kits for this yet. It originally came capsicum from peppers. So a lot of our vegetable crops are where all these viruses come from. If you have a vegetable garden and you grow cucurbits, peppers, and tomatoes, well, there's your 3 major groups that you could be introducing to your collection. All right, so now that we've seen what viral symptoms look like, what do some non-viral symptoms look like that are similar? You see that leaf right there where it's got that expanding margin of necrosis? It's not a ring, it's not a spot that's starting in the center of the leaf, it's moving down from the tip. It could be tip leaf burn from excess salts. It could be water was sitting on the tip of that leaf and a fungal pathogen took, and it's starting to progress down the leaf. That advancing necrotic margin, and you can see a little bit of spotting on that leaf. So there's some sort of fungal fruiting going on, fruiting bodies present. So it's fungal, most likely not virus. Another thing that looks similar to orchid virus is mesophyll cell collapse. So this is something that can happen, chilling injury to your orchid. So if there's water droplets on a leaf, and it's outside and it's cool, it can kill patches of cells. Sunburn kind of looks like this too. I don't know if this was sunburn or water that was sitting on the leaves that led to sunburn or chill damage. I think that's a Cattleya nobilis. Can't remember which is bifoliate and unifoliate.
Emilio, bifoliate.
Yeah. Sometimes insect damage looks similar to this. If you have a scale infestation, a severe Boistavol scale infestation, you don't notice it. Sometimes it's on the underside of the leaf and you start getting this weird necrotic spotting. And then you flip the leaf over and there's all these scales in a big cluster. Even long after you remove the scales, the tissue damage is still there on that leaf. And it might not be necrotic at first, but then it'll present later as necrotic and chlorotic. It's basically, if an insect is feeding on a plant, sucking, some of them piercing, sucking insects remove the chlorophyll as they're eating the contents of the cells. So like eventually, it's gonna present itself like a chlorotic spot. Could also be sunburn, like a lot of times during the summer that happens too. So yeah, this was scale. That's what the scale looks like.
Wow.
It's that, it was in that crease of the leaf as it was developing, there was Boistavol scale. And as they fed, they caused chlorosis. And at this point, the scale infestation has been controlled, but it's now turning necrotic. And it looks really bad, like a viral symptom, but it's not.
Christian, do you still find a lot of boys develop?
When I took over the collection, there was a ton of it. That one was a plant that I had gotten from someone else that had it.
Because in the 70s and 80s when I first started growing orchids, it was very common. I don't see it much anymore.
It's very easy to control.
Yeah, I see the brown scale more, but I don't see the boys develop.
It might be that in
And it's
commercial
pretty
production, it's been controlled more now.
Yeah.
It's mostly passed between people when you give someone a division of a plant. Very easy to control though. Like, I mean,
Although
oops,
it's
we're going the wrong way. Like I treated that plant with just insecticidal oil, and it was extremely easy to just control that colony. So nutrient deficiency. Nutrient deficiency can sometimes look like that too. It could be an iron deficiency, manganese, something that's not mobile. Is it phosphorus that isn't as well mobile? It's been a while since I've taken plant nutrition, but the immobile elements, if you're not feeding your plants a complete fertilizer, you can start to get nutrient symptoms, nutrient deficiency symptoms in your orchids. The media, there's nothing in it. It's bark and maybe some charcoal and some inert clay pellets. So this is what nutrient deficiency in iron would look like in an orchid leaf. Another symptom here is bacterial soft rot. So a difference between, well, this infection is pretty severe at this point. You can get necrotic spotting in your leaves, but it's bacterial. The difference is it's watery, water-soaked appearance. The tissue goes downhill really quickly, like so quickly that like one day it'll start as a little spot, the next day the whole entire new growth is down. That one is very easy to control using some sort of sanitizer in your collection regularly after irrigation. Some more virus lookalikes here. So leaf spotting, fungal leaf spotting on your leaves. We have Phylosticta and Cercospora there. It looks very much like viral necrosis because it's sort of venal there, but it's very pinpointed and it's on both sides of the leaves. And you'll usually, if you look closely, you can see fruiting bodies for most of the fungal pathogens. Sometimes you have to slice it open and put it on a slide on the microscope. But the difference is you'll actually be able to see something. Rather than a virus, you won't see the virus on your slide or on your dissecting scope. Here again we have a leaf that is presenting necrotic spotting, but it's pinpoint lesions. And it's not along those veins, the main veins of the flower. And it's the way it develops as well. Like, I mean, you'll know when there's moisture on the flower and there's like little condensation spots. The spores land on that spot. Overnight, this will start appearing. And it won't progress in the same way as the necrotic ORSV necrosis, where it follows those veins and then just expands quickly. Sometimes if you dry that flower out, like that's all it'll look like. It'll never expand. Color break in flowers. This is natural. I did not put the name under that flower. I should have. But every time this flower comes out, I'm like, there's something wrong with it. And all the flowers in the plant, they always look twisted. Half the color is on one side, and it's got that little bit of variation on the other. This is just what the flower looks like. It's not a virus. Every time I see a flower, I'm like, it's so underwhelming and ugly that why am I growing it?
Why do you do that?
I don't know.
Shocking.
It doesn't present well. It's like hanging off the edge. Like they're tiny, tiny little flowers. I wish I had the name of it. So how are viruses spread? And we can thank Jason for this slide with all the different vectors. So these are all well, except for this one. Your tools, your potting material, your pots, your clips, your hangers, like all that stuff can get dirty, have virus on it long term.
Communal watering sites.
Yeah, like this is dipping in a bucket. It's not a good practice for spreading virus.
They'll do it.
I don't know what kind of thrips that is, or what kind of thrip.
I don't know.
But some thrips can transmit virus, so control your vectors. All right, so to that tune, start clean, stay clean. It's easier if you start clean. So ultimately the best way is to prevent them from entering your collection and prevent them from spreading in your collection. Only buy plants from reputable sources. Some growers acknowledge viruses, some are like, they just happen, you get them. So you can return some plants to some vendors and they'll refund you, others not so much. They won't even acknowledge it. Segregate your incoming plant material. You get a new plant, don't be like me and just pot it up and put it with the rest of the plants. Keep it separate for a few months, for a while, until you test it. Or if you don't test it, just observe symptoms. Don't just place it with all the other plants. Like I said, virus testing all incoming plants is what I've started to do at this point, just because I don't have anywhere to segregate the new plants. Be diligent in insects and diseases in your collection, especially under the bench or like anywhere around where you're growing your plants. Weeds, 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 weeds. I think that, what was the one that was a host for? It's a Tritoscanthia type plant. Blue flowers, commonalities family, I can't think of it. But it's a weed. And it is a major host for, I believe it's like ORSV. Peppers, cucurbits, like any of those weeds, any of those families have weed species in that family that are common weeds. Like in the solanaceous, the tomato family. A lot of weeds are solanaceous, and they'll grow around your house, around your garden, and they can host CymMV and spread it to your plants. So keep things clean, weeds, pests. And then practice good hygiene. So like that's the biggest thing with CymMV and ORSV, is good hygiene. Like here on the bench here, we have multiple different methods of sanitizing. So on the left, we got the benzomatic torch. You can flame sterilize. That's one of those things that they are saying is not extremely effective for viral sanitation. Because when you think about it, you're only flaming one. You're not flaming your entire pruners, they have plastic handles. You're not flaming your gloves. Anything that the plant sap has touched can have the virus in it. You're only flaming a small section of the blade. It's kind of inconvenient heating it up. How hot do you need to make it? How long? There's different chemical sanitizers. Physan is there, one of them that you mentioned. That's another one that's not proven to be extremely effective against ORSV and CymMV because they're so stable. It's a quaternary ammonium product. You could use it for other things. It's great for bacteria and fungal pathogens. Still use it when you're repotting to like sanitize stuff, but don't use it as your primary defense against the virus. I won't go through all of them because we will later, but decisions, decisions, decisions. How do you decide what you're gonna do? Like, it's overwhelming when you see how prevalent orchid viruses are. The first step, I think, is to assess your collection. Like, what plants just don't look good? I think Fred Clark talked about this during the summer.
Mentioned that, yeah.
If you're growing a plant, and I have tons of these that I have, they just look terrible. Like, this has been looking, I'm like, oh, I need to repot it, I need to repot it. Maybe if I repot it, it'll look better. Why? Are you taking up space on something that hasn't bloomed in years? Like the last 3 growths. It looks terrible. Even if it didn't have virus, why am I keeping it? You've got that little piddly plant, that seedling that you think you can get to recover. Maybe it might just be a host
Although
for
if it doesn't have virus, you can bring it here and maybe someone else can grow it.
Yeah. Sometimes you just don't have the conditions in your greenhouse to grow it. But like, don't be afraid to throw things away if they don't perform well. That's the first step. Flowers with color break. That's an immediate, it's virus, get rid of it. You can't have it judged. Like, there's no point. The flowers are the reason you grow the plant. So that's an immediate kick it out of your collection. If there's symptoms showing on a plant, the next step, like you're assessing your collection, you're like, oh, that looks terrible. Is it a viral symptom? Segregate it, test it, see if it is a virus. So there only used to be 2 viral test kits commonly available for CymMV and ORSV. Those were the 2 most readily available viral test kits you could get on the market. Now, I mean recently, like, because this is like in the last year or 2, now they have them for an Agdia, which is one of the testing companies we'll talk about. They have them for cucumber mosaic virus, impatiens necrotic spot virus, potty viruses, which I think it covers 1 or 2 of the other viruses on that list. It's just a general group. Bean yellow mosaic virus, tomato ring spot virus, tomato spotted wilt virus. So these are all viruses that orchids can get, and they're commonly available from one company. I think they're all the same price as the old tests that were CymMV and ORSV. They're all just as easy. We'll talk about how those work. But it's all how far you want to go, because like all these tests cost money, and they're all separate, except for the CymMV and ORSV. You can test those together, but all the others are separate. I mean, how much plant tissue do you have at a certain point? Like, how much are you going to slice off and start macerating up to test? Like, so how you decide to test is up to you. It could be you separate out all the plants that don't look good, you test those. And then you practice really good sanitation in the greenhouse so you're not spreading anything that's asymptomatic to another plant that might express it. Some people don't treat viruses as serious as other people do because of sentimental reasons for a plant. They might hold on to something even though it's virus because it's historic and rare. Like it might be one of the few divisions of a plant left, and it's historic. They might be sentimental. You might have sentimental value. I have those plants too. Like you don't really want to get rid of them. Kind of just got to keep them isolated. You can have a separate area, separate grow area, and separate tools and stuff that you use for your virus plants. If the flower break isn't a problem, and the flowers look good when it blooms, if the symptoms aren't too debilitating, if it's asymptomatic, maybe you want to keep it. Just don't bring it around us. Don't give it to someone else unsuspecting. Just know that you have a virus. Should you test for orchid viruses? As I said, that's an individual decision. You could just go based on visual symptoms and whether the plant looks good or not. But when you're buying a plant for that much money, why wouldn't you spend an extra $450 to test it? And then if along the road that does show symptoms and it does go downhill, well, you lost your money if you didn't test it. If you tested it when you first got it and you contacted that grower, they might refund you the money and give you another plant that's not virused. Depends on what kind of grower they are. Some take it seriously, some don't.
$125.
I know, that was, believe it? That was when I was
naming names today. That's why we're naming names. Are they still in business?
I visited them in high school. They were out in like Indianapolis.
Yeah.
We're never gonna see them. I was just looking through old photos and I was like, I must have taken a photo of it because I was like, whoa, that's an expensive plant. But yeah, that's the same grower that they had virus in their greenhouse. That's a type of Phalaenopsis, I
It
think.
looks like a fail for $125.
I don't know what it was. I didn't take a picture of what it was.
Wow.
But like I said, some vendors take it seriously and will test in their collection. They might not and have indexed the entire collection, but they'll acknowledge it and refund you. Some won't. Okay, like they could test plants in a batch from a lab, but then somewhere along, it takes years to flower an orchid, somewhere along that growing process, somewhere along the retail chain, it could get virus. Even if it's certified, don't like go on that. Like it could have sat on a bench in a garden center and gotten virus. Someone could have been pruning or cleaning in that greenhouse and spread the virus around. All right, so virus carriers. Now this is courtesy of Jason. So this is one of his Phalaenopsis that was asymptomatic. It looks good. It looks perfectly healthy. There's nothing on those leaves that looked wrong. Did you notice anything, Jason, at
Absolutely
all?
not. So I buy a lot of plants. Don't tell my wife, although she knows. And so I think I've had like 2 dozen positives in the last year. 2 dozen. Not a single, maybe 3 were from plants in my collection that retested, and I think 1 was like not vigorous, so the cavity got sorted out. And everything else was on new material. And not a single one of the new plants I had showed any symptoms. No streaks, no color break, no nothing. This plant, this is not my photo with the flower, although it has since flowered. Looks completely fine. The vendor refunded it. I said, "You want it back?" He's like, "No." And I'm like, I couldn't pretty much throw it out because it's completely healthy. So I threw it, bring it to my work where it's a barren wasteland and it's very warm there. It's Phalaenopsis. It's blooming right now, and you can see it's florally positive. Not a single symptom, and so that's the scary part.
So, yeah, like when I did index the entire collection at work, I ran across a lot of plants that were really vigorous, and perfect, nothing wrong. I mean, the root growth was phenomenal. Tons of new growth, tons of new meristems.
You would not know. I mean, you showed great pictures of symptoms that you should really think if you see a plant that has like symptoms, it could be virus. But you know, it's shocking how much virus is in the commercial retail chain.
Yeah, I was even like, as I was testing, I was like, is this a false positive? Like, I'm getting so many of them. Like, how do I trust that these are truly positive? And I mean, true. Like, if this plant was really valuable, and it was part of like a historic breeding line for an orchid program, and you tested it with a lab, you get an immunostrip test, you'd send another sample to a lab to confirm.
I might set up seed pod on that. I mean, you know, it's you can set seed pod, dry seed, dry pod won't transmit virus.
Yeah. So.
You know,
Yeah, you can get, like I said, virus. Seed does not
green
transmit
pod,
virus.
Yeah, dry seed you can do.
Yeah, but it's how careful they are with cleaning the seed. That's important. I had a chart of like how many washes with sodium hypochlorite it took and whatever. But yeah, I mean you could use it for breeding.
I know commercial breeders. I mean, I know some people. I'm not gonna name that have a lot of virus and they produce tons of seedlings and they're all clean.
They just have really good sterile practices
Yeah,
in the lab.
or their lab that they use is clean.
So that, I mean, we haven't talked about the viral test kits yet, but this is what they look like with the immunostrip kit. This is another one from Jason.
So I this is actually the one, I'm not gonna name anyone, but I bought this Phonopsis and it was like, it was a nice stem prop. It was like, not cheap. I tested with Agdia. It was quite around there, a little bit less than that maybe. But I don't buy a lot of Phonopsis, but there's a lot of viruses in Phonopsis, unfortunately. I tested with Agdia, that's on the left. So the control strip's on the top, that's a control line, that's always positive. The medium one is ORSV, and you see it's faint, so it's kind of equivocal. And the CymMV mosaic is the bottom, and that's clearly positive. And then the vendor said that's a false positive, and I wasn't buying it, so I did a lot of research, not Facebook research, real research. But and someone sent me a Riga kit from the West Coast, and I did it, and it exactly correlated. I mean, you can see the Riga's flipped the other way. So the top is control, the middle is CymMV mosaic, which is clearly positive. It correlates exactly to the idea. And there's a really faint line, which again, I'm not sure this plant is ORSV or not. It's equivocal, but clearly the CymMV mosaic was
Yeah,
positive.
and this is
Not a symptom.
not asymptomatic and could have both viruses.
Yeah. Oh, this is unfortunate. So I bought this from a reputable vendor in the DC area, and I was like, this looks like virus. But he tested it before he sent it to me. I asked him, like, I don't want a virus plant, but this is a very famous cultivar. I tested the plant when I got it. It was fine. The leaves, and it bloomed like this. I'm like, this doesn't look good. And so he told me that it often does bloom like that. So I felt a little bit better. I tested the flower itself afterwards, just again, and it was like negative. At least for those two viruses. Could there be something else? I'm not certain.
Yeah, I mean, there's another
But
30.
it certainly doesn't look like it's ORSV or CymMV mosaic.
Another 20. But I mean, sometimes, how old of a cultivar?
This is from like 80 years old.
Okay, so like,
It could have something else.
yeah, it's likely something else. But also, like, if something is, plants don't live forever without their genes starting to get
Yeah,
affected.
yeah.
Like, this is a division of a division of a division of,
Yeah.
for 80 years.
Yeah.
Someone's been cutting this plant apart. And like, when you do that, it takes a lot out of a plant to live that long. The genes may have started to like be affected. Like, there may be just some genetic freak anomalies that are starting to affect how it flowers.
Or some might be prone to that. It is possible that some cultivars are
Yeah,
just prone
some
to
are prone to the breaking.
Yeah, yeah.
I mean, hey, they got some great, really cool petunias out there that they've been.
Well, there's an ansett. The ansett has a variety of like with streaks. It's like it's a very people like it. It's streaked, but it's really not virus.
Like that flower I showed with that weird coloration and distortion, yeah. So the two most common viral test kits out there are Agdi on the left here, and it's immunostrip assay test kit. So you macerate the plant material in this little pouch. There's a little buffer in there. Then there's a strip that contains the, what do you call it, the binder that'll bind to the virus that is potentially in here. It'll move up the stick and bind to another protein marker on the stick, whether or not it's positive. So that's one common one. Another one is Regabio on the right here. Slightly different. They're both very similar to like the COVID test kits. I mean, when those came out, I was like, oh, interesting. We could do it to people too.
Is there any guidance on what part of the plant to test?
So we'll go over, there's more info on how these test kits work,
Because I
but
think that's important, right?
yeah, so these are just the two most readily available tests. You can send plant material to a lab, but it's more expensive. In terms of pricing, we'll get to that, but it used to be like you could test for $4, and that was pretty reasonable. It's gone up since then. So these are what the kits look like. So these are the primary kits for CymMV and ORSV. It's really nice that they have kits that combine those into one strip. So you don't have to buy two strips and do two tests separately. Since it's similar in transmission, and they're just the most common viruses. These are the kits they made up. Last time I looked, the price had gone from $155 on Agdia for 25. No, was that for 125? Yeah, for 25 strips, it was $155, and just this past like 2 weeks ago, it was 170. So it's gone up. I think it's around $680 for a test kit. I think that was. I'm not sure if that was the old price with shipping.
That's the old price.
With
Now
shipping,
it's
the old price was $680.
$7.25.
Yeah, so now it's $7.25. Or if you don't have that many plants to test, you can buy them in kits of 10. You can buy kits of 10 or kits of 25. They're a little bit more expensive. They're $14 each if you buy 10. So there's a bulk discount the more you buy. We'll talk about at the end of the meeting, if anyone's interested in testing maybe a plant that's symptomatic, we can order bulk, like 25, 50 test strips, and you could buy how many strips you want individually. So you don't have to spend $300. I mean, from Regabio, they're cheaper individually, but you have to buy 50 of them for $200, and that $4 doesn't include the shipping yet. So it can, and that's shipping from Japan.
Taiwan. I don't
Taiwan.
think it's that much. Probably like 450. It's not.
$450, yeah.
It's not like terrible.
It's probably
a small
$50 for shipping. Yeah.
or less, maybe less.
Is there an expiration date on the strips?
So that is the other thing. So I think these last, so this was the most recent batch I bought in December of 2023, and these have a use by a January 2025. So they're almost 2 years shelf. These have to be refrigerated. The Agdias, you have to be kept in the fridge between 2 and 8 degrees Celsius.
But the Regabios are cheaper.
The Regabios are cheaper, and they don't need to be refrigerated.
Temperature.
You keep them at room temperature. I don't know what
They're,
technology is different.
They're individual pouches, and they have like one of those moisture, and a strip. Each strip is individual. I just used one today, and there's one of those moisture. You know what I'm talking about? The little
package
It's an
of
oxygen
moisture.
absorber and a
so
moisture absorber. Yeah.
Agdia is 25 strips in one.
Yeah,
jar,
they're all the strips are in a little canister.
which is fine, but if like eventually, I mean, if you put them in the fridge, moisture gets in, that will potentially warp your test.
Yeah.
Sounds like Regabio is a good idea, a good deal.
But this is a
There's
U.S. company.
There's a subtle thing. We'll probably get to it later. but
Yeah.
I batch test, so I can put like 6, 8, 10 samples in Agdia without overloading it. Regabio, you get a tiny bit, and you can get false positives if you use too much.
Oh, okay.
You have to look at like the data behind how accurate the tests themselves are. And I mean, the Agdia tests are pretty darn accurate. I mean,
I think Regabio, there's more sense. If you have a miniature, perfect. You only need a tiny little bit of tissue for the other one, the Riga or Regabio.
Yeah.
so
But you can't put that much in there.
yeah, we'll go over the procedure of how much tissue and like what you need to
It
test.
tells you.
But here's just the breakdown that Jason did.
That was
Based
before
on
they
the
raised the
old
prices.
pricing.
That was just 2 weeks ago.
That was 2 weeks ago, yeah.
And now I guess.
And I just literally, when I was making this
2024.
presentation, I went up and looked up
New
what
year,
the
new
price of the
price.
tests were. And I was looking for the CMV, the cucumber mosaic virus test. It's like, oh, they're $170 for a pack of 25. Okay. But they're a little more expensive than the RSV, and then I go look at the RSV, and it's going up too. Yeah, so these are just what it would look like, price comparison. There's no shipping, well, this is with shipping, this is without shipping. Moving on to how this procedure works. So I've only ever done the Agdia test strips for CMV and RSV. So you can take a sample from a leaf, you can take it from a flower, you can take it from a flower stalk, you can take it from a root. It just depends what you choose to. I'm not sure if there's any higher. The chances of it being in the new flower or something, I think maybe that's because it's new growth. I always choose old growth because it's been on the plant for longer. It's new growth sometimes grows so quickly that it can outgrow the virus.
Especially catasetums, I found false negatives, I suspect, because they grow so quick. If you take a new leaf, virus hasn't caught up to it yet. That's my suspicion.
Yeah, because like they can do meristem tissue culture in a lab to extract the meristem from an orchid that has virus and produce a virus-free plant. It's a process that, I mean, if it's really valuable for breeding, you could do that. It's not guaranteed to work, but yeah, the meristem of the plant is growing so quickly that it outgrows the virus. So I always test old tissue of a leaf usually. I've never tested a root before.
I tested roots. They're usually I like leaves better.
Yeah, it's easier to just slice off a tip of a leaf and to measure the amount of tissue you need. So step one, select your tissue sample. These are all the materials you need, well, I use to sample. A single-use razor blade. I don't want to use a pair of pruners between plants. I don't care how clean it could possibly, you could make it. I don't want the chance, like if you're testing one plant that's positive, and then you're immediately testing another plant of contamination. Single-use razor blades, I use an X-Acto knife. Super easy. The blades, 300 packs, they're like 20 bucks. That's all I ever use for deadheading in the greenhouse, is those X-Acto knives. So for a flower, a leaf, and a root, it's 0.15 grams. Now they tell you it's like the size of like, I think a dime or something, like the amount of tissue. But orchid leaves vary in thickness and density. Like a cattleya leaf, it won't take very much. If you have a Brasidium leaf, a Catasetum, like it'll take a lot more surface area. So you can't go by size, you go by weight. And honestly, there can be a margin of error to it. Like it doesn't have to be exactly 0.15. I'm like, okay, as long as it is 0.15 and then maybe up to 0.18. I use a jeweler's scale. I mean, on Amazon, the thing was like 18 to 22 bucks, they're cheap. And it's high accuracy, a little tiny sample plate here, and it's a tiny little scale. That's what I use. So I got the scale, I got the single-use blades, the test kit, the Agdia test strips, gloves, a pair of scissors to open the bags, because they come in a little buffer bag, and macerate the tissue. And a sanitizer. Even though I'm using single use, anything else that I'm touching, I want to sanitize my hands, the scale weigh boat, the bench. You notice everything's wet. Use a sanitizer to wet everything down. And then I chose a plant in the greenhouse that needed repotting. I pulled out my blade, and I took a little sample off the oldest leaf, or the oldest healthy leaf. And sometimes I kept a couple of pieces because I didn't get it right, but that was what I came up with, was 0.169 grams from a leaf. You can use flower spike tissue, but it's a lot less tissue, 0.03 grams. That's not much at all. You'd really have to weigh that in the scale to know you got 0.03 grams. Okay, so Jason talked about batch testing. So if you want to test multiple orchids and use one test kit, you can. You just use a small amount of tissue from each plant. And this is great if you don't want to test plants that, sure, they're not expressing symptoms. You think it's unlikely they have a virus. So you could test, I mean, up to how many, Jason, have you done?
I wouldn't recommend this, but I've done like 12.
Oh, geez, okay, that's a lot.
Because I just recall my paths and just take whole leaves and I just put them up. I have not seen overload false positive with Agdia, or nothing conclusive or reliable. You can't do that with Riga.
Yeah, so Brian, you mentioned this, that they do this at the internship you worked at, batch testing for orchid viruses. About how many would they combine?
Yeah, I hadn't heard before that you can overload the viruses. We did the test strips where you masticate the tissue in like a little vial. I'm not sure what
That's Riga probably then.
That's really good. They would come in those big boxes and break it open individual ones.
Yep.
I just remember they told us to get vascular tissue, so we'd go in to the side of the leaf and get it from there instead of the tip, and that's just how he went by it. For batch testing, we'd do 5 at a time when they came in, or less. It didn't really matter. The whole idea was just to save a little bit of money on tests. And of course, if the batch test came up positive, then you just go in and do individual tests for each one. So it's not always worth it.
Yeah.
But if it's a grower you get stuff from and it's clean, then you go into the batch testing. But it's like individual plants, and it wouldn't make sense to batch test them if they're all from different vendors.
Yeah. And about I've always wondered how much tissue. Like it's not much if you're doing 5 different ones, because you weren't worried about overloading them, so.
No, not really. I mean, obviously less tissue and there's liquid in there, but it would just be enough from each. When I say like a 1/4 inch by a 1/4 inch size notch, it wasn't anything crazy, but most of our collection was cattleyas and pathogens. So.
Yeah, because I've done most of these tests, and the only thing that can happen when you overload is that it gets really thick and syrupy. And I don't remember what orchids it was, because I tested everything in the collection, and a few of them the test just runs so slowly, and the test becomes the strip when we look at it became green, and it was really hard to read the results. That was the only thing that could happen. Like I said, single-use blade. That's step 1. Step 2, the sample bag. There's a buffer in the sample bag. You cut it open, there's a little mesh in there. You peel the mesh apart, you drop your sample in between the mesh, you can grind it up. You do it with a blunt object, so you don't tear the bag. The scissors are perfect, they have a nice sturdy handle. Macerate the tissue until you see pale green solution in the bag with the buffer. They advise that you let it sit for 3 minutes. I mean, I sometimes run a bunch of them at a time, so I start with the first one. Label it so you know which one it is. You're not going to remember if you put one down before the other, then you picked it up, they'll look the same. So make sure you keep track of the virus testing you're doing. Wait a few minutes until it settles out a little bit and everything is homogenized in the solution. And then,
And mostly for the bubbles, I think, because I had a problem with if you have too many bubbles and you're too
Yes.
over eager, you can short circuit the strip, and then you just wasted $7.
Yeah, so you don't, with the strips, you have to be really careful how you handle them. You don't want to just grab the strip anywhere. You can only grab the top of the strip, so you're not contaminating your results. Because at the bottom of the strip is where they have the protein binders. So they say to just touch it at the top. You want to make sure the bubbles are out of the way. There's a little channel built into the side of your sample bag. So after the tissue sample is homogenized, you slide your little strip in there. I usually squeeze the bubbles out of the way and make sure that I shake things around so there's a good amount of sample liquid on the right side of the bag. Slide the strip in until there's a little line that says sample on the bottom of the strip. And you don't want to insert the sample too far into the bag. Otherwise it won't wick and it won't grab onto those protein binders and wick them up at the right rate. You can, the strip, if you get this top section of the strip wet, it won't wick from the bottom anymore. So then, 1/4 inch where it says sample, keep the bag in upright position for max firm. It may take up to 30 minutes to run the test. You usually see it in the first 5 minutes. But you could wait maybe up to 30 minutes. You could see in this one there's a very faint line for the ORSV there. Sometimes it's a low titer sample, there's not a lot of virus in there, and you want to give it that time to let it run. So you can see there's a really long instruction there on the bottom there. This is what they give you for interpreting the results. It's similar to like a COVID test strip as it runs. At the top you have the control line. There's the ORSV test line is the next one down, and then the CymMV is the one at the bottom. You can see these are negative results here. They're giving you some examples. If the line is green and it doesn't show that red marker, these are negative. If it doesn't show the control line, it means that it got stuck somewhere along the way. That's a false positive. You don't want to use that.
Have you ever seen that?
I have never seen that.
control.
I imagine it would happen if you got the strip so wet that it didn't wick all the way up, and it just got stuck, or if you didn't have enough liquid and it got stuck, interpreted too soon. I don't know. So these are the 4 different states that you'll see on this test strip. This one has a control line, the ORSV and the CymV. This one is just CymV. This one is just ORSV, and then this one is negative. So those are 4 different results you could get with these test strips. So this is something that Jason brought up. There are some problems that usually it's not that the test is invalid, it's something went wrong along the way to the test.
I actually mentioned something about red flowers. I mean, I've tested red flowers. I've never had a false positive, meaning that you had a positive if I never, they mentioned it, but I hadn't seen that. I hadn't heard anyone who had seen that, but they mentioned it.
So there's a lot of different steps along the way.
But Riga is known. I mean, I think people have people have talked to Riga that if you put too much tissue, you can potentially get a false line. And that's the one where you can only put so much tissue in that little vial anyway.
So those are just some things to keep in mind. They give you really detailed instructions on the kit itself. That's just an overview of how it works. So for disinfection, these are the different all the different sanitizers. I've done some research, and there's a really good article on, I think it's Orchid Viruses or something. It's one blog that someone runs, and they did a really deep dive to what actually kills orchid viruses, what sanitizers work and what don't. So they're very persistent, these 2 viruses. And like I said earlier, it takes 100 times the dose of UV radiation to deactivate RSV compared to common bacteria. So like you think that just leaving something outside in UV light is enough to kill the virus. And in most cases it's not for these viruses. You could have something sitting on a greenhouse bench, a leaf, that has viral particles in it, and it'll, for 10 years, I mean, that'll still be viable. So the most common sanitizers that you can find are trisodium phosphate. So this one will elevate the pH. It's a good first step cleaner. A super saturated solution of TSP. The next one that they mentioned was lime, sodium hydroxide. That's another one that elevates the pH. Another one is bleach. So those 3 are the good first step cleaners. They're cleaning at a high pH. Other ones include Virkon S. This one's a viricide that's used in the animal husbandry industry. So agricultural sanitizer for like, if you have a pen house or a cattle barn, like these are used to hard surface disinfection with a focus on animals. There is a little section in the brochure about greenhouse sanitation using Virkon S. This is one that I just bought because it's come up multiple times. It's non-corrosive compared to some of the other sanitizers we're using here. So you could use Physan 20, it's quaternary ammonium. Green Shield, another quaternary ammonium. They're the same exact product. One is labeled for use on hard surfaces only. The other one is also for plants. Not much of a difference. I don't think any of the active ingredient is different. One is just a little more expensive than the other. There's Xeritol, and it's a parasitic acid cleaner. This one, I didn't run across any of these parasitic acid cleaners in the research that I had done, where they were testing how effective they are for viruses. I'd assume they would be because they're extremely corrosive. And I used to use this one commonly in the greenhouse. And if you keep your pruners wet with that all the time, they're going to rust. If you kept them in a jar, your pruners dissolve. So I'm pretty sure that one's going to work for viruses too. If you drop it on living tissue like your hands, it's kind of like hydrogen peroxide, except 10 times more concentrated. It'll eat through your finger. So that one is one that it's used commercially. I think I'd given some to Mary in a bottle once at the Hartford Flower and Garden Show. And it leaked. And it was all over my hands. My hands turned white because it ate through all the skin on my hands. And then my hands peeled. Yeah. So always wear gloves.
Yeah. So the Virkon S might be a
So Virkon S is a safer alternative that I'm going to try out. I just bought a container of it. So the first step is using that elevated pH cleaning and then sanitizing with Virkon S. Physan 20, they're saying these quaternary ammoniums aren't great for the second step. So I use bleach. I use TSP and bleach as my two sanitizers. Flaming tools, really, it isn't great for sanitizing. It only sanitizes at one point that you heat up. Like, there's a lot of other points of contact with that plant than just the cutting edge of your pruners. You put the pruners down, there's a little bit of virus on the table. You touch the pruners, you got plant stuff on your hands, you got the virus on your hands. I like to wear just cheap nitrile gloves between plants. You could protect your hands, you could dip them in the sanitizer solution, your hands are clean. I mean, I coat everything, the table, the pruners, the supplies, the trug that I'm pre-potting in with the sanitizer. You really want to make sure that anything that has gotten contact with the plant is sanitized. So, best practices. It's mechanical transmission that we're trying to limit. When you work with your plants, anything, pots, potting mix, stakes, clips, hangers, pruners, work tables, benches, hands, crates, buckets, watering implements. There's anything you can think of. The mouse that walked through your collection and chewed on one leaf and chewed on another leaf. So you see here, like, this is a nice bench because you can wet it down and sanitize it. It's a hard surface, non-porous surface. Some people, they pull out paper, they use newspaper, they use butcher's paper because it's waxed and holds up water a little bit better. You want to change out your work, either change out your work surface between plants, or clean it really well between plants. Make sure that you're using a sterile tool when you're deadheading. Don't just like break a flower off or take your pruners and cut one flower and walk over to the next one, cut that one off. That is a guaranteed way to transmit. I use the sterile single-use blades. Or if you're going to sterilize your supplies, I even don't trust it to that point. If you're cutting into living tissue when you're deadheading, that's such a small little task that you're doing, just use a single-use blade for that. If it's dead tissue like a flower stalk that's thoroughly dead, you could use your pruners and sanitize between. But you might nick the plant at the same time, so just be careful. Good sanitizing protocol is important.
And spraying with alcohol really doesn't do anything.
Alcohol does not, because those viruses are so stable. That's likewise saying Physan. You think quaternary ammonium products are enough to get that virus, and it's not. You want 99.9%. You want 100%. So I did a great deal of research and talking to people, and like you start hearing what people are using. TSP, I mean, it's used in the research greenhouses up at Cornell. When they're working with their plants, using trisodium phosphate. It's really like heavy duty stuff. You just want to be careful you're not polluting the environment with it at the same time, because it's phosphates.
Is it toxic?
It's
to us?
harder to find now,
Is
actually.
it toxic to us?
I don't believe it's toxic to us, but I mean, it's such a high pH that when you're touching it, it's extremely slippery. And I mean, imagine it's slightly caustic to living tissue, so wear gloves. You're using a super saturated solution is what you want. It's a solution that like, at the point where like nothing dissolves in the solution anymore. There's crystals at the bottom of your solution. It's just a really high rate. So the first route, single-use razor blades. Like I said, that's what I use when I'm deadheading. X-Acto knives are great. You could use the U-line blades right here. Something that you get in bulk and it's cheap because you're throwing them away. You're using them once. They say you can sanitize them in the oven. I mean, you could put them in the oven at 400 for like an hour, and sure, it's probably sterile at that point, but like,
No bother.
they're cheap enough, they rust so easily. Like you can see, I didn't even finish this pack, and a little bit of moisture in the greenhouse. The last few blades are getting rusty at that point. Just throw them out, just dispose them safely. The other thing, for everything else you use in the greenhouse, I use a two-step process to make sure that 99% of the viral RNA is deactivated. Like you just want to be as clean as you possibly can be. So, I always make sure we're using gloves, first step. Put on a pair of gloves. You're protecting the plants and you're protecting yourself from all those cleaners. Next step, I use Xerotol as my second line of defense. I always mix up a 1 to 50 and put it right on the bottle so I can never forget how much I need to make 1 liter of Xerotol at 1 to 50. So that's the highest rate it's labeled for use for. Sometimes I'll just double it, but the thing about Xerotol, Sanidate, Oxidate, those are 3 products, same active ingredient. The thing about that is that you have to mix up a fresh solution every time you make it. So like, you can't let it sit. It's peroxide based. It's gonna disintegrate in water. It's just gonna turn into oxygen. It'll degrade over time. So I keep it in spray bottles. I keep it in a little dispenser bottle. I have a little measuring tube. Very small amount for such a small little spray bottle. It's cheap, easy, you can carry it around with you all the time. Most gardens have started carrying around a spray bottle with you. Out in the landscape, spray down your pruners between plants. Other plants get viruses too. That's my first step. The second step is the dip. I use the cleaning at an elevated pH. Sodium hydroxide or trisodium phosphate. Those are 2 that work well for that. And so the first step, I'll wash the pruners down to remove gross debris with the Xerotol. I don't want them to stay wet with Xerotol because it eats the pruners like crazy. They get rusty immediately if they stay wet. So I wash them off because you don't want to be dumping all root particles and dirt, contaminants into your solution. And then I'll put them in the TSP for 5 minutes or more. And so I looked it up. The solubility of TSP is 12 grams in 100 milliliters at 20 degrees Celsius. So that's just room temperature. The maximum solubility of TSP is 12 grams in a liter. It's easy. Use a mason jar. If you wanted something deeper that covers the handles too, you could do that. Just mix it up at the right rate. So then the next step is that oxidation step. Another 5 minutes. In this case, I'm using bleach because I didn't have Virkon S yet. They sit in the bleach or the Virkon S for another 5 minutes. So while you have pruners in production line, you have a pair that you're using, you have a pair that's sitting in the first step, you have a pair that's sitting in the second step, you have multiple pairs so that you're not waiting around because it takes time to sanitize. 10 minutes is usually what they say for 100% decontamination. So when repotting, this is the procedure that I came up with. And like at first, I wasn't doing all this, and I was getting viruses spreading in the collection. Before I even touch the plant, I'll test now. Just because I know I have it in the greenhouse, even after testing and throwing out all the plants that had virus in the greenhouse, some plants are still getting it. Somewhere there's supplies, a bench, some source of it in the collection, buying new plants. Wasn't 100% when I tested the first time, I don't know. So I test now, just to make sure that when I'm cutting the plant apart, I'm not gonna spread it to the next one. If you're not working with a lot of plants, it's not a big deal. Like if you're only working with 1 plant and then you sanitize, it's pretty easy. But if you're working like 20 plants at a time, like you gotta make sure it's clean between. After a plant is confirmed clean by the virus test, it's only CymMV and ORSV. So I wanna make sure that I'm going through all these other sanitation procedures between so I don't spread any of the other viruses. But like I shake off all the loose media, dunk it in the Physan 20. It's great for bacteria and fungi. So I'll dunk it in a Physan 20 dip at the labeled rate. I think it's like a tablespoon or 30 milliliters per gallon. While the plant is soaking, I make sure the work surface, all that debris from depotting is all over the table. So I clean the table, spray it down with Xerotol at 1 to 50 rate. So the table is clean now. Only pruners and knives that have gone through that decontamination come in contact and cut that plant. Make sure that the materials you're using to repot, the pot, the clips, the media, are all clean. Don't reuse your media. Don't reuse the pots and clips unless you've sanitized them between uses. It's harder with a clay pot because plastic pots, they're hard, non-porous. You could easily scrub them clean, sanitize them. Clay pots, it's a little bit iffy. I mean, everything I grow is in clay, so like some people say throw them out. I can't do that because I can't find them to buy them. But I make sure that it's contact time. With any sanitizer, it's contact time. So with bleach, I use a 10% solution, and 10 minutes isn't enough for a clay pot. But it goes through green shield for 10 minutes, which is the quaternary ammonium. Scrub it clean, and then it goes into bleach overnight. I'll leave it soaking in a 10% bleach solution. And I have another slide about the life of sanitizers, but over time, sanitizers decay and they're less effective. But you can get that long contact with the sanitizer, overcomes it, and it's enough to the point where that pot is clean enough for me to use again. And then the last step I use is Xerotol alkaline just to spray down everything, including the plant, just so that everything is clean. So like I said, the bleach decays over time in efficacy. If you get a 10% solution, it's not going to be 10% an hour later. It's going to decline. Green Shield is one of those quaternary ammonium products like Physan or Green Shield. Those are more stable. You can keep the solution as long as it's not excessively dirty and overloaded with organic matter. It's still effective. Bleach becomes less effective over time. So make sure you scrub off that gross debris. Use a sanitizer like Physan or Green Shield for 10 minutes. That'll get all your fungi and bacteria and other pathogens. And then overnight soak in bleach is what I use for the clay pots. It's as good as I can get. I mean, some people even bake their pots in the oven at 400 for 10 minutes. And I mean, when you have several hundred pots, I don't think I want to put that procedure through my oven. They use autoclaves at research greenhouses. Like back at Cornell, everything that went in and out of a greenhouse was autoclaved. And I mean, they had big, gigantic autoclaves that you could walk in. And they were loading things in with forklifts. Even the plants that came out of the greenhouse, after they were done with the research, would get autoclaved. Make sure you killed everything.
Wow.
Nothing would survive that. And so, thank you. That's all I've got.
That's a lot. That was a good presentation.
If you want to come up and look closely at the orchids that have symptoms, we have plants that are symptomatic here. You can take a look. If you want, we have 2 plants here that some people brought in, and we can virus test those if you want to see how the test happens in person. Otherwise, enjoy the show table if anyone has the bravery of, well, it's 9:15 at this point, but if anyone wants to go through the show table with Dave and us, we'll be over there. Yeah, sorry.
Okay.
Tired.
I was terrified.